all our attendees. You can add any questions to the Q&A box throughout the seminar. If you wish to ask your question yourself, you can raise your hand and we'll unmute your microphone. So thank you again for joining us, Anna, and take it away.
Thank you. Thank you very much, Connor. Thank you all for attending and for being here. I'm going to try to share my screen now. Where am I? Here. Share. Okay. Can everyone see my screen?
So now you're in presenter mode for us, Anna.
Okay, great. But you can see the screen or the presenter mode?
Presenter mode, yeah.
Oh.
So I think if you go to the three dots next to the camera icon, you can switch. Next to the arrows with the slide number.
Give me 1 second.
Or swap displays, that should do it too.
Swap displays. Yeah.
Swap displays, right.
Here.
Okay, that's good. That's it, yeah.
Awesome, fantastic. Let me see if we can move this in here. Okay, perfect. Okay, great. So where am I? And you can see my pointer right now?
Yes.
Fantastic. Great. So yeah, thank you all for being here. Thank you, Connor, for the nice introduction, and all the EpIC seminar organizers. Thank you, Leila, Osama, and Jelena as well. It's a real honor to be invited to give an EpIC seminar. And as Connor just mentioned, I just started my position in Colorado. So what you're going to be hearing today is the last work that I did during my postdoc in Kenton Schwartz lab at the NIH. So I hope you are a KV channel aficionado, and if not, I hope by the end of the talk you will be, because there's a fascinating channels to study. So as the title of my talk says, we're going to be talking mostly about inactivation mechanisms in voltage-gated potassium channels. But and I just want to mention that I'm going to be talking mostly about slow inactivation in KV 2.1. And this is a story that has been already published. But at the end, I do want to give you like a little teaser of the last story that I work in Kenton's lab on fast inactivation or N-type inactivation in a classical voltage-gated potassium channel that is shaker. But before I go into the mechanism, I do want to go back to the very early days of the field. And I do want to talk a little bit about the experiment that Hawking and Huxley did back in the 50s in the giant squid axon, where they describe the conductances that model or that form the action potential. So as you know, they describe two different conductances. Sodium conductance that activated and inactivated within milliseconds. And this is the first time that inactivation was used to describe an ionic conductance. And the way we understand it today is exactly the same way they described it back in the day, as a decrease in the ionic conductance while the activating stimuli, which is voltage in this case, is still present. They also describe this delay potassium conductance that show after the sodium one, and within the time scale that they used for the experiments did not inactivate. So what they didn't know at the time, but we do know now, is that these beautiful ionic conductance that describe the action potential are actually carried by ion channels. And I'm going to be talking about my favorite channels in the world, voltage-gated potassium channels, that I am fascinated to just understand how they work and which mechanisms regulate their function. So as you may be familiar with, they are one of the most big families of in the human genome, like families of proteins in the human genome. They're very important physiologically. They are involved in many physiological processes like cardiac function, neuronal firing, hormone release, immune response, you name it. At the cellular level, they are kind of like the gatekeepers of the resting potential. They are responsible for bringing the membrane potential back to its resting level after a cell gets activated. So you can imagine how, because of their importance in these physiological processes, that point mutations in these channels will produce that very detrimental syndromes like cardiac syndromes, neurological syndromes, autoimmune diseases, etc. So they're also very interesting protein targets for pharmacological companies to produce drugs and small compounds to regulate and modify their function. So I think this audience do not need this in this slide, but just in case, just to have everyone on the same page, just want to go very briefly over the basics of KV channels. So a classical KV channel is a homotetramer. Each monomer contains 6 transmembrane domains. The S1 to S4 comprise the voltage sensor domain, being the S4 the segment that has the positive charges that will sense the changes in membrane potential. The movement of that voltage sensor domain will be transduced by this S4S5 linker that covalently links the voltage sensor domain to the pore domain that is formed by the segment 5 and the segment 6. So upon changes in membrane potential, the voltage sensor domain will move up upon the polarization, as you can see here. That up movement is going to be transduced through this S4S5 link into the pore, opening this region of the channel named the gate, and that will allow potassium ions to flow into the gate, get coordinated in this region called the selectivity filter, and then flow outwards, producing these beautiful hour rectifying currents that you can see here. We can actually plot the current obtained in the current trace as a function of the voltage pulse that we gave, and then get these beautiful sigmoidal curves that will reflect the voltage dependency and the open probability of the channel. Now, these channels can undergo other states after they open, and this has been described for shaker, which is this classical voltage-gated potassium channel. And they can undergo inactivation. But there is 2 types of inactivation described. There is a fast inactivation that has been modeled as a ball and chain mechanism. And that fast inactivation is characterized by an activation within the millisecond timescale. So you can see here how the channel opens up on the polarization, and within milliseconds, it inactivates and goes silent. Now, if we remove this N-terminus of the channel, this like ball and chain, genetically or by proteolytic enzymes, then you can see how within the same time scale, the channel does not inactivate anymore. However, if instead of milliseconds, we give second pulse longs, then you can see how the channel can undergo another type of inactivation called C-type inactivation in shaker, because the region of the channel that was responsible for this slow inactivation was towards the C-terminus of the channel, was actually in the selectivity filter. And we just recently saw how that looked like, and it's like a dilation of the selectivity filter. But why do we care about inactivation, and why is this important? Well, because at the physiological level, it is important. And what I'm showing you here is a synapse between a CH3 pyramidal neuron and a MOSI fiber. And you're seeing here a train of action potentials that looking like just from the, like in this perspective, does not look like much. But if you superimpose all these action potentials, now you can see that from the first action potential to the 100th action potential, there is like this widening of the action potential shape. And what that produces is actually an increase in the calcium current in the presynaptic bottom. And eventually, what that will produce is an increase in the excitatory postsynaptic current. So this is producing synaptic facilitation. And as you can probably guess, the reason why this first presynaptic broadening of the action potential is happening is because of KV channel inactivation. So with that, I'll go into the first part of the talk, talking about, in this case, slow inactivation in KV 2.1. And as you may be familiar with, KV 2.1 is a fascinating voltage-gated potassium channel. It's a delayed rectifier. It's very much like the one that Hawking and Huxley described back in the day. So you can see these beautiful delayed rectifying currents as response of depolarizing voltages. It's very voltage dependent, and of course, it undergoes slow inactivation. Just so to make clear, this channel does not have fast inactivation. It only has slow inactivation. However, we know that the slow inactivation in KV 2.1 is different than the one that has been described for shaker. And that is because permutations in the pore domain that speed slow inactivation in KV 2.1 in shaker has no effect in KV 2.1, is insensitive to TEA, and the effect of extracellular external potassium does not have the same effect in shaker than in 2.1. So it's very similar to Shaker in terms of the architecture of the channel. However, there's many functional things that are like intrinsically different, and we did not understand how they work. And we're fascinated about this channel because besides the things that we don't understand about it, it's a very interesting channel to study because it's highly expressed in the brain. So you can see Kv2.1 here highlighted in green in the sagittal cut of a mouse brain. So you can see how it's very widely expressed. It controls the neuronal firing in some neurons. So this is like dopaminergic neuron from the substantia nigra. So you can see the firing of the action potential and how after adding GXTX, which is a very specific toxin for Kv2.1, you can see how the frequency firing gets faster, and eventually the neuron gets depolarized and cannot fire action potentials anymore. And more interestingly, point mutations in the gene that encodes for Kv2.1 cause epileptic encephalopathies and developmental delays in the kids that carry them. And these are a group of neurodevelopmental disorders that present epileptic seizures within the first year of life and are unresponsive to like classical treatment. So we were fascinated about this channel. We want to understand more about it. So we saw the structure of Kv2.1 using cryo-EM, and it was like giving us the same architecture that we've seen for other potassium channels that we saw before, like for example, Shaker. It was a homotetramer, voltage sensor domains in the periphery. So it was very similar, but it gave me like a very interesting roadmap for me to like put all these or like locate all these disease-causing mutations in the channel. So what you're looking at here is the structure of a monomer with the voltage sensor domain here, this first 5 linker, and this will be the pore domain with the S6 helix highlighted in color in blue. So you can see different disease-causing mutations that I characterize and that are located all over the place. And as you can see, they present gain of function, they present loss of function, they're all over the place. But the one that I want to talk about today, and that I'm going to be focusing for the rest of this part of the talk, is this phenylalanine 412-2-aleucine that is located at the bottom of the S6 helix. So if you see within the tetramer, it's like right down there looking towards the voltage sensor domain of this subunit, kind of like pointing away from the permeation pathway. Here in the zoom in, you can see how it's kind of looking also towards the pore domain of the neighbor subunit. And you saw before that I showed you traces for the other disease-causing mutations, but I didn't show you traces for this one. And the reason why that is because I couldn't get ionic currents with this mutant. I only got gating currents. And that was fascinating because a gating current is the reflection or is of the gating charge movement across the membrane electric field in response to different voltages. So you can see here how upon depolarization, you can observe the on-gating currents and the off-gating currents upon hyperpolarization. And if we integrate the off-gating currents, we can obtain the QV curves that show the movement of the charge as a function of the voltage. And that was fascinating because we are now mutating this phenylalanine to aleucine, and now the channel is not able to conduct ions anymore. So I was very puzzled by it, and I wanted to understand how this point mutation is actually making the channel non-conducting. So I came back to the structure, and I find out that around that leucine, this phenylalanine, there was 3 leucines that were kind of like coordinating this like phenylalanine in this kind of hydrophobic nexus. So all these residues were hydrophobic, and there were kind of like this hydrophobic interaction distances. So I thought that was very odd, and I then like went ahead and mutated all of them to alanines to see how they were all interacting together. So I'm just going to show you, for the sake of time, L403 and L316A, but if you are interested in knowing how this looked like, let me know, because I do have that at the end too. But basically, when I mutated those leucines individually to alanines, I observed, like, first of all, that it looks like within the same time scale, they inactivated a little bit faster than the wild type. And when I plotted the voltage dependence of activation of these mutants, you can see how L316A does not change the voltage dependence of activation much, but L403A does shifted to more depolarizing potentials, suggesting that this mutation is probably stabilizing the closed state as well. So this was very interesting. Like, now I have mutations within this region that if I mutate this to a leucine, makes the channel not conducting, but mutating these residues to alanine also affect inactivation and the closed state of the channel. So I was wondering, okay, is Kv2.1 being able to inactivate through some mechanism related to closed state? In other words, is Kv2.1 able to undergo closed state inactivation that has been described for other Kv10s like Kv4s, for example? So in order to test inactivation a little bit better, I did these like classical 3-pulse protocols where you give like a first pulse to a known voltage to assess how many current you have in this you have to begin with, and then a second pulse to different voltages, very long one to allow the channel to inactivate or reach steady state inactivation, and then a third pulse to the same voltage than the first one to see how many channels did actually inactivate during the second pulse. And what you can see here with these long 3-pulse protocols, L316A inactivates faster than the wild type, like 10 times faster, and L403A inactivates even faster than the wild type or the other mutant, like 100 times faster. And if you see the scale, I didn't even need to give like the 20-second pulse protocol that I used for the wild type. For this mutant with 5 seconds, actually with 1 second, you have enough. So I was really intrigued by this because it looks like the residues in that hydrophobic nexus were both stabilizing the closed state, were speeding inactivation dramatically to the more dramatic phenotype, which will be the Phi412L, which is the disease-causing mutation and is not conducting ions. So if the closed state inactivation was the mechanism that was being like coordinated in this nexus, there was a classical potassium channel blocker that could actually help me kind of like untangle that mechanism. And that was for aminopyridine. So this is a very classical non-specific potassium channel blocker that does many things, but it's also an FDA-approved drug for multiple sclerosis. So they give it to the patients with multiple sclerosis because in the brain, it blocks the potassium leak in the demyelinated axons. So it helps restore the conduction. So it helps with the motor syndromes of the patients. But at the voltage-gated potassium channel level, it stabilizes the activated and open state, so it stabilizes the pre-open state, but it also competes with this closed state inactivation. So if the mechanism by which Kv2.1 is inactivated and these residues are modifying the mechanism is closed state inactivation, 4AP should compete with it. And that's experiments, those are the experiments that I did, and this is what I saw. So you can see the same 3-pulse protocols that I used to study inactivation in absence before and in presence after I add 4AP. And you can see how in the mutants, 4AP is slowing inactivation dramatically. It's competing with it. And it's also happening in the wild type channel, which is telling me that probably this is the mechanism that Kv2.1 is using to inactivate. So this was great, but if this is the mechanism that also controls the non-conducting behavior in the disease-causing mutation, if I add 4AP to that mutation, then I should regain conduction, right? Because probably the channel is inactivated through this mechanism. And that is exactly what I saw. So when I add 4AP to the disease-causing mutation, I can regain ionic conduction. And when I wash it, then I go back to not having ionic conduction anymore. So this was great because we found this hydrophobic nexus in Kv2.1 that seems to be the responsible for the inactivation mechanism in the channel. And we'll explain why other manipulations and other things that were happening in the selectivity filter trying to use the same mechanism that we had for a shaker for Kv2 didn't work. It was because it was happening somewhere else. So now we wanted to see how this inactivated structure looked like. So we tried to solve the structure of this mutant using cryo-EM, the same way that we solved the structure for the wild type, and it didn't work. It didn't work at all. We tried many things. We tried a lot of different variations biochemically, it didn't work. So we went for the next best mutant, which was L403, because L403 is inactivated faster. So we thought we had a chance to solve the inactive structure with that mutant, and we were successful. So we could solve the structure again using cryo-EM. Overall, the structure that we saw for L403 was very similar to the wild type channel. voltage sensor domains in the periphery, in the activated state, selectivity filter coordinating potassium ions. It was all very similar except for the gating region. So we saw the structure using C1 symmetry because L403A was an asymmetric structure. And that asymmetry within the pore region presented this constriction around the isoleucine 405 that was actually non-compatible with the permeation of potassium ions. So we thought like we solve actually the structure of the inactive state. And what I'm going to show you now is a morph between the wild type structure and the mutant. So you can see within the mutant in these two structures how these two S6 domains are kind of undergoing this rotation and exposing those side chains to the permeation pathway. And don't worry, it's going to play again, but you can see the hydrophobic nexus here. When we introduce the mutation, what happens is this hydrophobic nexus kind of like breaks down. It's coordinated in the wild type, but when we mutate residues, that it breaks down. So you can see this 2S6, and you can see how in the L403A mutant structure, those side chains for the S6 are kind of rotating and occupying that permeation pathway. So just so you can see kind of like a screenshot of the wild type versus the mutant, this is the S6 helix, and you can see how now the side chains are exposed towards the permeation pathway, producing that kind of constriction. And the hydrophobic nexus here is actually broken, so the phenylalanine is kind of like falling off the nexus. And of course, when we mutate the leucine to alanine, that moves that S6 too, which is the one that is exposing the residues. So this was great, and now, but we wonder, because we know that these residues in the hydrophobic nexus are highly conserved in other KV channels. So we wonder if this was also conserved in other KV channels. So we did a structural alignment, and we saw that this hydrophobic nexus, it is present in other KV channels. So it's a mechanism that may be available for these channels too, although it may not be the one that they use for inactivation, but it's kind of like right there and it's conserved. And we also wonder if it was conserved in other voltage-gated ion channels, like sodium or calcium channels. And I just want to take a step back before I go into the conservation on other KV, on sodium channels, to the very early experiments that I mentioned to you, derived from Hawking and Huxley, where they describe the inactivating conductance on sodium channels, on sodium conductance, sorry, and how then Clay Armstrong did this beautiful classical experiment where he treated that squid axon with a pronase, a proteolytic enzyme. And you can see how after the pronase treatment, the sodium channel did not inactivate anymore. I'm going back to this because that is why they proposed the ball and chain mechanism, like Clay Armstrong and Pancho Manzanilla proposed this model a few years later, where an intracellular part of the channel will physically block the pore, and that will stop the ionic flow on the channel. So this was the model that it was adopted for sodium channel inactivation. And the intracellular inactivation particle was later described as this IFM motif by the residues that form them. And the model was that this IFM motif, this inactivation particle, will actually block the pore and stop the sodium flow. And this was like the way we understood this mechanism until 2018, when the first structure of a sodium channel in the presence of the IFM motif was solved, and they found that the IFM motif was not being coordinated in the pore of the channel. It was actually being coordinated in this side pocket between the voltage sensor domain and the pore domain of domain 3 and the pore domain of domain 4. So if you're seeing where I'm going with this, when we superimpose the structure of Kv2.1, this hydrophobic nexus, with the structure of the sodium channel with the IFM motif, we found that the AFM motif was being coordinated in this same kind of hydrophobic pocket that we described for 2.1. And the phenylalanine of the IFM was like occupying the same spot that the ring of the phenylalanine of the Phe412L was occupying. So that was very interesting, and that was showing us that probably these channels use a similar maybe not the same, but probably a similar mechanism to inactivate. And actually, recent work by Pancho Ventanilla's group show how inactivation in sodium channels also involves residues, which are behind the pore in this region here. So with that, and actually this slide is gonna help me a little bit with the introduction for the next part. I'm just gonna go into the last part of the talk, which is our latest results in the fast or in the N-type inactivation, this time back to the classics in Shaker. So as you remember from this slide from the very beginning, the fast or N-type inactivation in Shaker was described as a ball and chain mechanism. And this is because if you think about it, in the 90s, when they first cloned Shaker and they saw this kind of behavior, the classical idea in the field was like, well, this is like a fast or like fast inactivation, like the one what we see in sodium channels. Therefore, let's see if it's like the same mechanism, and let's ask the same questions, and let's kind of like break the mechanism the same way. And I just want to bring up this quote from this paper in 1993, which is like, I think, a funny one, because it says like, they thought like back in 1993 that N-type inactivation was the best understood gating transition in Shaker, but here I am, 2025, talking to you about N-type inactivation again. So hopefully I can convince you that there is something else to learn about it. But going back to the classical experiments, of course, if you see that fast inactivation behavior in Shaker, the first experiments that you are going to go towards is like a proteolytic enzyme, kind of like the same one that Clay Armstrong did with the pronase. And in this case, the group of Rick Aldrich did these experiments using trypsin. So you were looking at here at single channel recordings, it's like the closed state, and those are like bursts that reflect the opening of the channel. So you have 1, 2 channels in response to a plus 20 millivolts depolarization. So you can see how the channel opens and after it goes silent, which is like because the channel is inactivated. But after the trypsin treatment, you can see how the open probability is much more, is much higher. And you can see that in the single channel level, but also you can see in the macroscopic current in the control before and after trypsin. And after this, they went into this quest of identifying the inactivation peptide by doing several deletions in the N-terminus of the channel. So you can see the different deletions that they try and how the open probability look like in the single channel experiments. And here you can see how it looks in the macroscopic current. So you can observe how different deletions will change the inactivation behavior of the channel until they got into the infamous Shaker delta 646, which is probably one of the most or the highest used construct in our field. So when people talk about Shaker inactivation remove, they're probably talking about the 646 that has a deletion from the residue 6 to the 46 and the channel does not inactivate. Now, later studies by Rob McKinnon and Rick also show how there is only one N-terminus required to inactivate the channel. And this is like a very interesting paper when they use toxin sensitivity and the availability of different N-terminus to assess how many particles we needed to inactivate. And they found like only one, although you will have always 4 available because it's a homotetramer. There's only one that's needed to inactivate the channel. And then later work by Gary Yellen also showed that the recovery from the N-type inactivated state was both voltage dependent and was potassium dependent as well. So in order to do these experiments, they did these 3-pulse protocols where they did like a first protocol to a voltage to allow the channel to open and inactivate, then a second pulse to different lengths, and that you can also change the potential. So you can use different recovery voltages to allow the channels to recover from the N-type inactivated state. And then a third pulse to the same voltage than the first one to see how many channels recover during that pulse. And you can change the potassium concentration. So you can see here how increasing extracellular potassium speeds the recovery from N-type inactivation. And the mechanism that they propose, or the model they proposed for this, was kind of like in a charge repulsion model, similar to the one that has been proposed before for positively charged toxins. So increasing potassium concentration will kind of push away that N-terminus from the gate. And on top of that, they also describe how the mechanism by which the N-terminus will be blocking the channel was actually an open channel blocker. So the channel needed to open to then get blocked. And then during the recovery from that N-type inactivated state, the channel was allowed to open, therefore showing that the port had to be open during the recovery. Now, but what is the structural identity of that N-terminus? So there is like a very old structure of just the N-terminus alone that crystallizes a beta sheet. So is it a beta sheet? Is it an alpha helix? Is it an unfolded peptide? So that was not clear. And there is like this paper by Miguel Holmgren where they use MD simulations to actually simulate what will it look like. Will it be like a beta sheet, an alpha helix? And it looked like the unfolded peptide will be the most likely. Also, it will fit with many, many papers that did functional data trying to ask that question as well. But we didn't have a structure and we didn't really know how this will play out. So that's where we thought that was a very interesting question to ask. And Xiaofeng Tang, which is like my partner in crime in all these projects, took the challenge and decided to solve the structure of Shaker in an N-type inactivated state. So I'm just going to give you like bits and pieces of the work. So stay tuned for the paper. It should come like in the next months. But just to give you an idea of all the structures that he solved multiple structures using cryo-EM, where he saw no peptide at all, different conformations of the peptide, different locations, different degrees of blocking on the selectivity filter. So you can see a full engaged peptide here. And you can see, I'm just going to go to this one here, this beautiful structure where he saw like a continuous density from the T1 domain all the way up into the selectivity filter. So I'm just going to be focusing on this one right here, which looks like this. You're looking at the pore domain of two subunits, the S6 helix of two subunits, and this is the unfolded peptide from the alanine 2 to the residue 12. And as you can see, we have enough resolution to solve and identify some side chains. We see how there is a continuous density with I470, which is this residue that we know is very important for N-type inactivation. There's like a lot of work done to question, identify like this residue here. And as you can see, besides all the side chains that we see and the different interactions that I will touch a little bit later, I am showing you that we solved in the structure, the alanine 2, we didn't see the first methionine. So we were wondering what was going on here and why are we missing the first residue? We're not seeing the methionine and we're actually seeing the alanine that we know it can interact and cross-link with I470. So this looked like it was right, but we were missing a residue. So we took the same sample that we used for cryo-EM, and then we did that with all the samples that we use for cryo-EM. And I showed you before, every sample we used to solve a structure, we send it for mass spec and see what was going on there. And we repetitively saw that the first methionine was not detected. We detected just from the second alanine on, and you can see like the relative intensities here, the absolute intensities here. So the methionine was not there, and the second alanine was actually acetylated. So most of the alanines that we had in our sample were acetylated, showing that we were actually having like a post-translational modification that happened in the cells. So the cells were doing this after the protein was being created. And this is actually something that we were not aware of, but it's something that happens very regularly in mammalian proteins and actually protects the proteins or shields from degradation. So to determine whether this truncated and acetylated channel corresponds to the channel that we have been recording since forever, like functionally, I went ahead and took this protein, the same one we sent for mass spec, and the same one that we use for cryo-EM, reconstituted into liposomes and inject the liposomes into the oocyte, hoping that those liposomes will fuse with the membrane and bring that protein, that purified protein, to the membrane of the channel, so I could do functional studies with it. And I was very shocked, but it worked. And as you are looking at here are the currents that I recorded from these oocytes injected with liposomes containing the acetylated alanine shaker protein. And to my surprise, when I compare the kinetics of inactivation of these reconstituted proteins with the kinetics of inactivation that I obtained from just injecting the RNA into the oocyte in the regular way, you can see how the recovery, the kinetics of inactivation are indistinguishable at low extracellular potassium and high intracellular potassium. So it's probably the same protein. I also did this recovery from inactivation experiments with using the same protocol that I showed you before in the Gary Yellen papers. And you can see how the recovery from inactivation is, again, indistinguishable from the RNA wild-type injected protein in the oocytes compared to the purified protein reconstituted into the liposomes and injected into the oocytes. So this was very good, but now we have a problem. And is that I told you that the mechanism that we were thinking that the high potassium will speed the recovery from inactivation in these channels was due to this charge repulsion. But we just saw that there is no charge. We solved the structure, we also did the MS, and the residue that is closer to the selectivity filter is an alanine and it's acetylated. So there is no charge there to justify this mechanism of charge repulsion. So we were thinking, what's going on here, and what will be the mechanism that could be behind this potassium dependency on the recovery from inactivation, N-type inactivation. And then we remember that there is another mechanism that happens in the selectivity filter that is highly potassium dependent, which is slow inactivation or C-type inactivation in shaker. And we know from Gary Yellen's work lab that N-type inactivation promotes slow inactivation by blocking the exposure of the filter to high concentration of internal potassium, so that otherwise the gate will see when the channel is open. So therefore, we hypothesize that rising extracellular potassium and a negative voltage will speed the recovery from N-type inactivation by increasing the occupancy of the selectivity filter and pulling channels from the C-type inactivated state into the conducting state. And that will actually help the channel to recover faster from N-type inactivation. So in order to test that, what I did was to measure the same, do the same experiments that I showed you before, but now I was allowing the channels to enter C-type inactivated state by giving like a much longer pulse. So before I was giving 60 millisecond pulse, right now I'm giving like half a second pulse. And if this was true, if our hypothesis was true, then in low potassium, which favors slow inactivation, the recovery from fast inactivation should be slower and less complete. And that is what I see. So you can see how the fraction of channels recover and low K when I allow channels to undergo C-type inactivation is less complete. That when you have high K or when I do the experiments with shorter protocols. So you can see this is the recovery in low K with shorter protocol, 60 milliseconds. But when I increased the length of the protocol to allow the channels to undergo C-type inactivation, then the recovery is less complete. And of course, high K, because high K competes with slow inactivation and takes channels from the inactive state to the conducting state, that actually helps the recovery from inactivation much faster. So then the next thing that I did, because we were wanted to ask more about the relationship between C-type and N-type inactivation, was to introduce T449. So T449V is a mutation in the selectivity filter. So it's like here, it's like this turning here, that stabilizes the open state and slows down C-type inactivation dramatically. So if C-type inactivation is involved in the mechanism of the recovery from N-type inactivation, then with this mutant, it should be faster than in the wild type, regardless of the potassium concentration, right? And that is exactly what I saw. So what you're seeing here is the same protocol for the shaker containing T449V in low K and in high K compared to the wild type. So this is the fraction recover for the wild type channel, and this is the fraction recover for T449V, where I am not allowing the channels to undergo type inactivation. So you can see how the mutant is significantly less sensitive to extracellular potassium concentrations. And therefore, we conclude that C-type inactivation of the filter probably stabilizes the N-type inactivated state, and rising extracellular potassium recovers or speeds the recovery from N-type inactivation by pulling the channels from the C-type inactivated into the conducting state and helping them recover. And just in case you were wondering, I also tested T449B does not affect the inactivation kinetics, just in case you were wondering about that, because I did wonder about that. So just to finalize, I just want to give you like a few screenshots of the other contacts that we found out with these like new structures. So of course, in the lower section of the gate, we have leucine 7 very close to B478, which is another very well-known residue within the gate that helps helps open and close the gate of the channel. P475, part of the PVP motif, critical proline for channel function. Also lowering down the leucine 7 is close to this asparagine 482, and we see in this structure how the asparagine 482 is really, really close to this tyrosine 8, and those are all residues and things that I tested, and that will be included in the paper as well. And I just wanted to show you here that you can see how in blue we have this N-terminus, but we solve actually other N-terminus that are going to be around, because if you remember, we have 4. Only 1 blocks the channel, but we could see densities for other N-terminus around the gate as well. And just a little bit more of this density, the residues that we identify. And just to finalize, just to give you a little bit of a model that we envision more kind of a nunchaku mechanism, kind of like the N-terminus sneaking into the gate. And we solve several structures with a fully inactivated state, so we could see the non-polar interactions that happen within the gate. We also found that the N-terminal methionine is deleted, and the alanine on the N-terminus is acetylated, interacting with 470. And we find out that rising cellular potassium destabilizes the N-type inactivation by altering the conformation of the ion selectivity filter in the external pore, pulling channels from the C-type inactivated state and helping them recover from the N-type. And with that, I just want to thank deeply everyone in the Swartz lab. Thank you very much to Kenton for all the mentoring and the opportunities to do these beautiful projects. Special shout out to Xiaofeng Tan. All the structures you saw today were solved by him. He is, as I say, my partner in crime in these projects that we love. And thank you to Jensen also at the NIH for his support and his help solving structures. And Chan Hoon, who was a postdoc in the lab and was involved in the very early stages on the first project on the KB 2.1. And with that, thank you all, and I'm happy to take questions.
Fantastic. Thank you very much, Anna. Well done. So we've got a few questions in the chat, so I'll read them out in order. So the first question is from Paul Wagner. What enzymes could play a role in the N-terminal removal of the methionine?
That's a great question. Yeah, so there is and I don't want to say this wrong, but acetylases. There is a lots of them. There is a lot of literature on them, and that was fascinating to us because we were not aware that was happening. And from many other channels and proteins, we may not care much. We do care about the N-terminus in this one because of the mechanism of N-type inactivation. But apparently 80% of the proteins, mammalian proteins, get post-transcriptional modified and get acetylated. They will be cleaved in the first one, like the MAA is kind of like a target sequence for these enzymes. So they could be cleaved in the first one, and they could be also cleaved in the second one. And in some of the samples that we sent for mass spec, we saw that the second alanine was not present either. So we have samples from the third one. But the majority of the modifications that we observed in all our samples were cleavage of the first methionine, acetylation of the second alanine. And that was kind of like we found that it's like re-engineer. Let's go back again and try to think about this again, because this was something we're not aware of, but it's really common, and it's a really interesting modification.
I think that methionine aminopeptidase that removes the methionine,
Yeah.
I think the activity is somehow correlated with the size of the second amino acid, right? So if you substitute that alanine to something bulkier, you might interrupt that methionine removal. And does that change inactivation at all?
That's correct. That's a very good point. And we did not mutate in that alanine. I know people have mutated to cysteine to do cross-linking experiments, and that had other side issues and other things. And I'm trying to think if other mutations within that position specifically. But I don't remember. I know that they mutated charges in the N-terminus, like position 12 and 13. If you introduce positive charges there, then you change the speed of inactivation. So that's like a modification that people have done. And that's something that if it has been replicated also with free peptides, right? Like, so they will like also add free peptides and try to mimic the inactivation. So those modifications were done. The second one I know, but I do know for the literature, sorry. My apologies. From the literature on post-translational modifications, that if the second residue is not an alanine, then that methionine does not get cleaved.
Right. Let's give you a break to talk. I'll throw it over to John. I'll alternate between the chat and the hands in the room. So John, take your time with the question.
All right. Hi, Anna. Just all of this, it's like all marvelous things revealed that have been going on in these channels, and just to really to give the structural form of that to them, coupled with the dynamics and testing and retesting and testing better the hypothesis, the studies that led to these hypotheses in the first place. It's just cool. It's like looking at the world where you can truly see. I love it. And also with that, I had one kind of question, or even speculation, or maybe it was in there and I kind of missed it. But do you have a sense or a guess of what it is about the C-type inactivated conformation itself that stabilizes the N-terminal peptide in there?
Yeah, that's the question. That is the question. Yes. Yeah. First of all, thanks for your work. This is great. And you're right. Like, we're kind of revisiting all this like classical knowledge and finding out like things and new angles where we can understand a little bit more. So it's really rewarding, and it's super fun too, because there's so much good data produced that you can evaluate your data right away and say like, does this make sense? Does that make sense? And that is great. But your question is the question. It's like, how is that change in the selectivity filter affecting the N-type inactivation and stabilizing it? And then switching the equilibrium into the conducting conformation and speeding the recovery from inactivation, because that's what potassium is doing. That is something that is like a question. Like, we think it may be an allosteric regulation, which means that it's not clear how is it doing it, but something architecturally has to be happening for that to stabilize it. And actually, it's funny because the T449B experiments were the last ones that I did in the lab, like the last ones. And that was with that idea in mind of like, if the potassium is really through C-type inactivation, then this mutant has to be behaving in a different way. But you can see here how even, and this may be a data thing, but even with the mutant, there is still a slight difference between the 2 millimolar K extracellular and 100 millimolar K extracellularly. So it's not perfectly overlapping, which is telling me that there may be something else here, which may be related to the architecture of the selectivity filter and how that's happening, because the mutation is here, but we see the dilation happening from here, from the middle out. So there is a lot of things moving in here, and how that is affecting what's happening down, I'm not sure. It may be the potassium, but it looks like it's more related with the C-type inactivation.
Are there differences in the occupancy of the S0 site or the vestibule for the potassium ion in C-type inactivated channels,
Yeah,
right?
so we don't see anything. Like the only two sites that we see occupancy are like down here. Just these two. Yeah.
Yeah. Could it be just
Could
that even without the fixed charge of the amino terminus, just that the displacements by the potassium that can flow only when the filter is open is doing it all? I mean, that has its own voltage dependence.
Yes, that's a possibility too. And I just want to mention that these structures were solved in low potassium and low ionic strength. So these were solved in 4 millimolar potassium and 46 millimolar sodium, so 50 millimolar total. And that is based on, again, classical work by Rick Aldrich, but he also showed that the ionic strength will, like lower ionic strength speed in activation. So we were trying to modify these biochemical condition as much as we could to help solve these structures and help solve, like, see everything that we could just to get an idea of how this was actually happening. But yes, we decreased the ionic strength, we mutated the peptide, we also added free peptide just to kind of jump the conditions with inactivating possibilities, so we could actually solve the structure of the inactivated peptide. But yeah, that exact thing that was exactly happening here, I guess TBD. The best I can tell you is it's probably C-type, but how? Have to keep digging.
It's good. It's a good job you've got your own lab now. You can work this out. I'm going to move on with the chat because we've got a few questions building up. So a question from Damien Bell. Lovely talk. Thank you. The acetylating of alanine in the fast inactivation studies is fascinating. You mentioned it happens quite often. Do you know if the alanine in the L316A or the L403A mutants is acetylated as well?
Yeah, good point. We haven't, the short answer is we haven't sent that for mass spec to see if they have any modification, but I will think that won't be the case because those alanines are in the C terminals, within the channel, so they are not available for acetylation unless the channel gets cut there and that residue gets accessible. But I would say the first time that we sent these samples to mass spec was in this project and because of what we found.
A question from Morat Oz. Are these currents recorded in cut open oocytes or classic 2-electrode technique?
Yeah, thank you for that question. I didn't mention that. That's a great question. All the experiments that you've seen here have been done in classical 2-electrode voltage clamp experiments, injecting RNA, except in the ones that I did liposomes, and those were liposome injected. Yeah, thank you.
Colin Nichols says, very nice talk, Anna. He has 3 questions. I'll ask one, Colin. I apologize. We'll come back at the end if we can. Number 1 was, if 4AP is trapping the open non-inactivated channel by blocking as a potassium analog, how do these channels now show conductance? Apparently as big as the non-block channels.
Yeah, so it's because 4AP is not a great blocker for Kv2.1. It's a channel blocker, and let me see if I can go real quick there. But it's not a great one. Actually, if you see the concentration that I have to use for 2.1 is in the millimolar range. It's outrageous. It's a lot. It has many other effects. It makes the channel, the oocytes leaky. It has issues. But what I can show you, and I think I have it here. So what you can see here is in presence of 4AP. And these are H infinities obtained from the 2 mutants. Sorry, this is a 3, not a 1. In absence And in presence of 4AP, what you can see is like with 10 millimolar, I can block like 50% of the current. That's the maximum I can block. And the other effect, but the effect that I'm interested about is how that 50% is now seen to not being able to inactivate that well. So it's a very dirty drug. It's not great, but for the purpose of asking the question of like it is competing with inactivation, it was good enough. It was not great, but I would say it was good enough. But yeah, thank you.
Thank you. Have you got a few more minutes, Anna, if we keep going through the questions?
Yeah, please.
Okay, great. So another one from an anonymous attendee. Do you have any insight into how silent subunits such as KV6 interact and influence KV 2.1 inactivation?
To this here. That is a great question. And that is something that I know that it's many groups now going into silent subunits, like John is doing that and other people too. And it's a great question because we do know that it modifies voltage dependence of inactivation. I think we were going to go into the classical question of like, what's the stoichiometry 2231? But something that is clear here is that these are domain swap homotetramers, or sorry, heterotetramers with a silent subunit, sorry. But the domain swap is the key. That means that that hydrophobic nexus that it's in here and coordinating inactivation that has a part of one monomer and has a part of another monomer. We're talking that this hydrophobic nexus could be from the heteromer. And that could be how the silent subunits may be operating and changing the inactivation behavior. So that's something that I would love to see. And if people is thinking about it, that's great. But I think that would be my guess. And I would love to see that data. So if no one is producing it, let me know because I would love to do those experiments too. But yeah, that's how I think they will be doing.
The next question is from Harley Karata, and he mentions that you might have already answered this partially. Do you see classes of particles indicating N-type inactivated channels that are also C-type inactivated selectivity filter conformations?
Yeah. So I'm gonna go right real quick into this, and I know this is not a very detailed, but we solve structures of the channel in high K and in low K conditions. The high K conditions had the selectivity filter conducting, but because we know that high K will also speed inactivation, we switch right away into low K. Low K, and as I mentioned before to John, like low ionic strength. So most of the all the structures that we solve with the different N-type inactivated peptides in different stages, they all have the selectivity filter into a C-type inactivated state, just because we kind of like force it and wanted to go into that to being able to see more of the N-type inactivated or more stages. Yeah.
Thanks. Next question from David Colhoun. It surprises me a bit that voltage-gated channels have never been as analysed at the single channel level as ligand-gated channels have been. Experience with the latter suggests there will be more conformations than you suggest, than we can see so far, right?
Yeah, definitely. I mean, with these channels, we are kind of like functionally, we're blinded by what we can see and what we can record with experiments, but we know that there could be multiple other states in the channel. Actually, with 2.1, there is this beautiful gating models by I was blanking the name right now, but it will come to me. But when they propose how the channel can undergo multiple closed states into a final open state, and they can also inactivate from the closest state. So there's multiple closed states, close inactivated states. Unfortunately, we only see when we're doing ionic currents, the closest state that is closer to the open state. So we are kind of just like in that small range, but if we can use mutations and different things, probably we can unveil other states that happen that otherwise are invisible to us.
Thanks. Robert Stewart asked a question I was wondering about. He says, great talk. You talked about the conservation of the hydrophobic nexus in both KV and NAV channels, suggesting common mechanisms of inactivation. I'm curious if you have insights on what is responsible for the dramatic differences in the rates of inactivation between these channel types, if they have this shared mechanism.
Say that again?
Do you have any insights into the difference in the rate of the fast inactivation between the KV channels and the NAV channels, if they're sharing this common mechanism?
Right. Yeah. So nav channels are faster, are more effective into inactivating than potassium channels. The mechanism is slightly different. So so we know that these channels have, like, for example, for sodium channels, you only need to move 3 voltage sensor domains. The moment that the fourth one moves, it inactivates right away. And one of the reasons why we thought that this may be conserved and we may not be related that much with the rate is because of mutations that have been done in sodium channels that are not in the IFM motif. And I'm thinking about the CWC mutation, which is like a tryptophan scan that people have done in sodium channels. And they introduce mutations in domain 1 and domain 2 in actually in places where that are in the nexus. Actually, one of the positions is that leucine 403. So if you introduce a tryptophan there, and this is like a very well-known mutant in sodium channels, you actually slow inactivation dramatically. How that was explained without mutating the IFM motif was kind of a mystery. But for us, it made a lot of sense, because if the inactivation is being regulated outside of the filter, kind of like in the outer part, then those mutations will have an effect, and that's how it looks like. Now, how the efficiency of how inactivation happens in sodium channel versus Kv2.1 channels is like, I would say, is very different, because Kv2.1 is very slow. It needs seconds to inactivate. It actually, even within 20 seconds, you always have like a food. So there is never like a complete inactivation in 2.1, whereas in sodium channels, they're like super fast and super efficient. And that may be related with the role that they have to play in the action potential and how they have it to be that way. But yeah, like the coordination for the FM motif is like down there. And now the later papers from Pancho and Zanilla lab show that also residues there kind of like help to the constriction of the pore to stop ionic permeation. Suggests that it may be conserved, again, not the same, but similar, and kind of like the backbones may be there.
Thanks. One last question in the chat from Esteban Suarez Delgado. Greetings for the great work. Thank you for sharing. Bezania's group lastly showed an AlphaFold model with a cavity for 4AP that doesn't imply directly the pore. Do you think that the mechanism of competition with inactivation could be through an allosteric mechanism? Or since it's not so potent, you could have more than one binding site?
Yeah.
And then, other naive and technical question about injecting liposomes in oocytes that I was wondering about as well. How do you control the direction of the channel as it integrates into the membrane?
Right. So to the first question, yeah. So 4AP, we don't think it binds in the pore. We think it binds in this hydrophobic nexus, like right behind of the membrane of the pore in the membrane. It's actually a very hydrophobic drug, so you can put it extracellularly and it crosses the membrane and finds its way. So mutagenesis work from the 90s show that in channels that have high affinity for 4AP, like Kv3s and Kv4s, the binding site may be in that hydrophobic nexus region. They did that by mutagenesis. And one of the changes is that leucine L403 in Kv3 is a methionine, and that seems to be determining like part of the affinity for 4AP. So it's not the way that we think it's working. And if you think about it, 4AP has this like kind of like ring small structure. And the way we think about it is like that ring may be getting coordinated where the phenylalanine should be. And that is the mechanism by which the Phe412L will somehow regain conduction, because now you have this like, oh, let me see if I can show it. Like you can see like to have this small molecule that has this ring that whenever we're in the structure and we are removing that ring here, if it's getting coordinated here, it may help the coupling to happen again. And now I'm not showing it here, but all the residues that you have here kind of lining this area are all hydrophobic. So this is a very hydrophobic area where 4AP could be kind of like moving and getting coordinated and getting there. So yes, we think that that's what's happening. It's not happening in the pore, it's happening actually here. And then the second question was, can you repeat that?
Yeah, and when you inject the liposomes into the oocytes, right? The orientation of the channel.
Yeah, that is a great question. And that was part of the fear that I have going into those experiments and kind of the magic too. What when I started doing the experiments and I inject the liposomes, I track those oocytes every hour after I inject them to see how they were looking and if they were incorporating the liposomes and how they were doing it. So a first test that I did was to do two types of protocols, a depolarizing one and a hyperpolarizing one, to see if I have different orientations within the membrane. Now, we're doing TVC, 2 electrode voltage clamp. So we have a huge oocyte, and the injection, we don't know how it's fusing. We don't know if it's localized. We don't know if it's like more global. We cannot control that. But what I found is that all the currents that I recorded were all outward currents. I never saw inward currents whatsoever. So I'm not sure how the oocytes are doing it, but it looks like they have an orientation preference, or the ones that get incorporated like in the other direction are not functional. So that was a very interesting thing that I think the oocytes did for us that were like really nice. But in all the time that I was tracking those oocytes since I inject them until 24 hours after I inject them, I was only able to detect our currents that correspond to shaker.
Guessing the different composition of the lipids in the two leaflets might alter the channel behavior if it's one way or the other.
It may, yeah. We use the same composition for the liposomes that we use for the nanodies that we use for the cryo-EM, just to have everything within the same conditions. And they're basically a mix of POP, POPG, POPC. And somehow that, the oocytes incorporated them in a way that they were like just giving us like perfect currents. That was great, yeah.
We've still got 60 people online, so I'll go back to Colin's questions if you don't mind. So question number 2 was, if the one-fold symmetric inactivated channel occurs because of only one subunit with the mutation, do dimeric or tetrameric channels with a single mutation show similar activation rates and extent? Or does the single mutation act in a dominant negative fashion?
This is for the 2.1, I'm assuming, right?
I think so, right?
Yeah, no, that is a great question. And this is something that was great for us in terms of like understanding the structure and what we got, because this is a homotetramer. So all the mutations, all these subunits have the mutation. So yes, it could be possible that if you have only 2, or you have a concatemer and you have one mutation in 1, 2, or in 3, you may get the same structure. We model we model a symmetric structure. We model all the S6 having the same conformational changes. And we found that if all the S4s rotate as much as this one, and we see rotation in only 2, as I showed you in the video, only 2 of the 4. But if all of them rotate as the most rotated one, which will be D here, we will have structural clashes. So as if that was not allowed. Now, on the flip side, if you think about it, maybe only 2 are enough to inactivate the channel, and that's what the channel wants to do, because it's easier, it's cheaper energetically to just move 2 subunits instead of moving the 4. And we do have evidence that even potassium channels that are very synchronized, they're also kind of like asymmetric or asynchronized, because we do know that the voltage sensor domain can move independently, and only the final the final step that has to be concerted actually opens the gate. So this was kind of like puzzling, but also kind of reassuring in terms of saying we are not introducing structural aberration or something because we have a mutation, because the mutation is in all of them, but only 2 of them are moving and giving us an inactive channel. So yeah.
And Colin's third question was, how does the acetylated N-terminus even get into the pore? How does this compete with external K? I think we've kind of addressed that question. I bet Colin's thinking of the N-terminus of KIR 6.2 goes and binds somewhere in the sulfonylurea receptor, and we don't know how. So it's kind of a mystery.
Yeah, it is. But as I say, like the first part of and this is a common thing in all the channels, all the K channels that have inactivation through this type of mechanism, like BK2 and things, like the first part of the N-terminus is very hydrophobic. The residues are not exactly the same, but it's very hydrophobic. And some of them have then some charges. And we know that those charges kind of like come close to this S4S5 linker. So, and if you change those charges and mutate them, do you increase the affinity for the on rate? So there is a few things there that kind of like help getting the N-terminus into position, how the very hydrophobic N-terminus will plug into the board, I'm not sure. I will feel like they will try, like it's there, it's kind of snorkeling. It goes in between the T1 and the channel, so it kind of snorkels there, and somehow it finds the way. Maybe it's all the hydrophobic interactions that kind of favor that in an otherwise more aqueous environment. But yeah, that's a great question. Yeah.
That's cool. Yeah. I've got one last question, sorry, if I may. So
Yeah,
you
please.
mentioned that the hydrophobic nexus is observed in other KV channels that maybe aren't considered so slow in activating, like KV4 maybe, right? So are there disease-causing mutations in that region in any of the other channels that might point towards physiological or pathological implications of that mechanism?
Yeah, that is a great point. So on the top of my head, I'm not aware of other mutations exactly in that hydrophobic nexus in other channels, in other potassium channels. But I did find point mutations in that region in sodium channels that are related to disease. So it seems like a very important region of the channel to like have an effect and have like a profound effect at the physiological level. So yeah, but yeah, it's a lot of things to like learn here and a lot of things to explore.
Fantastic, and lots of interest clearly. So thank you very much, Anna, for the talk. That was
Thank
fantastic.
you. I'm gonna
And
stop
thank you everyone
sharing
for attending.
right now. Yeah, thank you all for the questions. They are great, and yeah, thank you very much for attending. This has been a blast.